Yeah, I had to, my Wi-Fi's been a little wonky this morning, so I just reset my router and hopefully all will be well. It's good to see you. If you haven't already, go ahead and type your name into the chat for me so I have a record that you've been here. That would be great. Okay. Very good. Thank you. All right. So today we're going to have sort of an informal lab meeting. We're going to be talking about the coronavirus a little bit. We're going to talk about testing methods for the coronavirus. If you recall, we talked a little bit about this virus earlier in the semester. We talked about how it's built, how it's structured, and how it gets into the body and what cells it binds to and so on. So today we're going to talk about how we test for it. You all have been through this sometimes uncomfortable testing process where you've had a swab put up into your nasal cavity. It can be very uncomfortable for some people. So you know that those tests are sent off to determine whether or not you're carrying the coronavirus up in your nasal passages. Some of you may have had those swabs evaluated by what's called a rapid test. We'll talk about that today. Some of you may have your swabs sent off to a laboratory for PCR testing. Remember, that's polymerase chain reaction testing. And we'll talk about that today. Some of you may, or maybe you know someone who has had some antibody testing done. Particularly early in the pandemic, people who came down with this virus and recovered were then asked in some situations to donate their plasma, donate blood, and then have the plasma portion, the fluid portion of the blood, prepared so that it could be given as a treatment, as a therapy to other coronavirus patients, because their recovered bodies had produced antibodies. And those antibodies in the plasma are one of the ways that we can treat coronavirus infection. So you may have experienced some of that. You may know people who've experienced some of that. And we'll be talking about those technologies today. So before we get into that, though, I do want to touch on some scheduling things that are coming up. We are rapidly coming to the end of the semester, as you know. This week, we have next week, which is the week of the 25th, I think. I'm trying to read my calendar. And then that's it. And then we're finished with the course. So we are rapidly coming to a conclusion. So I do want to pull up our Blackboard page because I want to show you an announcement, actually 2 announcements that I've sent out in the past couple of days, just in case you haven't seen them yet. Let me get this out of the way for us. So I'm on our Blackboard page here. That's what you should be seeing on your screen. I'm on the announcements page. I'm just going to scroll down. This is an announcement that I sent out on the 16th, just as a reminder about what's coming up in the last 2 weeks. The date of the final lecture exam, which is essentially the last event of the course, is Friday, April 30th. It's important to know that that is the final exam. That's the last exam for the course. It is not a traditional final exam, though. It's not cumulative. It's essentially just your next lecture exam. It's going to cover lecture topics that we've discussed since the last lecture exam. So the immunology topic, vaccination, epidemiology, and the last topic, which is coming up next week, which is called pathogens and how they spread. So it's going to look just like the other lecture exams have looked in the course, the same length and so on, the same types of questions. And it's going to cover this last set of lecture topics. The other thing that I posted in this announcement was that I have extended the last day that I will accept late work. I've extended it to Wednesday, April 28th at midnight. So 2 days before that last exam is the final date and time that I'm going to accept late work. So work that you, for some reason, were unable to submit earlier in the course. I decided to extend that date because I recognize how difficult of a semester this has been for everyone. I know that these are unprecedented times for us. Living in the pandemic. And I know that some of you have been dealing with family members who are ill. Some of you have been ill yourselves. Some of you have had to deal with quarantining, and I know everything has been difficult. So I'm going to do my best to make it possible for you to get credit for as many assignments as possible. So you may submit any of your missing assignments, if you have any missing assignments, up until Wednesday, the 28th, which is a week from today. I will grade those for you, and that will help boost your earned points and help improve your grade. Going back to the announcement page now, I did send this announcement out this morning. This is just a little bit of mathematics for you if you find it helpful. Obviously, there are a couple of things that are not submitted yet. If you've kept up with all of your assignments in the course, you know there are 3 more lecture quizzes to complete for the course. There's the vaccination quiz, which is due tonight by midnight. The epidemiology quiz, which is due Friday by midnight. And then the last lecture quiz, which is next week, Wednesday by midnight. That's the pathogens and how they spread lecture. So those are 5 points each. And then that just leaves that final lecture exam, which again is happening a week from Friday. And that final exam is worth a little bit more than the other lecture exams in the course. It's worth 100 points. So if you add up all of the points that have been available for you in the course, it comes in at 768.5 points. So that includes everything that was worth a point value for you to earn this semester. And what I've done here is just pulled out for you each of the letter grade categories, and using the percentages that are printed in the syllabus, I've got the minimum point value that you need to earn to get that grade. So it's still a little confusing, I know, because there are a couple of things that you have submitted to me that I have not finished grading. For example, I can see your question about the final draft of the pathogen assignment. Some of you have handed that in and I've graded it for you. If you haven't handed in your final draft, your revised pathogen assignment, go ahead and submit that to me. Remember, that's worth 15 points. And again, I will accept that up until next Wednesday. I'm happy to extend that, okay? You're going to use your first draft that I sent back to you, that I wrote comments on. You're going to use that first draft and the comments to revise. to rewrite and submit for me. And that final draft is worth 15 points towards your grade. So again, if you haven't submitted it to me, go ahead and get that in. And if you have submitted it and you haven't gotten your grade yet, try to be patient. Those will be done this week. The other grade that is outstanding is your unknown microbe identification. Again, some of them have been graded. I have graded and returned about half of them, but the other half will be graded this week. So keep an eye out for that. I will grade it for you and I will return it to you with my comments on it. I've been very pleased so far with the submissions that I've received from students. It looks like most of you worked really hard on that, and I think that you'll see that that's reflected in the grade you receive for that. Okay, so just to summarize, there are what I would like everybody to do is go and look at your grades on Blackboard. Open up your grades. Look to see if you forgot to submit anything, or if you didn't get to submit it because you just haven't had time to work on it. If there are any missing things in your grades, assignments you have not submitted, please go ahead and submit them. You have until next Wednesday to do that. So even if it's very late, I'm still going to accept them, okay? As long as you get them to me by next Wednesday, the 28th, okay? Now, I may have to take off a point or two for late submission, depending on how late the assignment is, but you'll still have a chance to earn the bulk of the points. So again, I really encourage you to take advantage of that opportunity if it applies to you. Some of you have, all your work has been submitted, so that doesn't apply to you. If you have grades missing in your list of grades because they haven't been graded yet, things like the final draft of the pathogen project or your unknown microbe homework assignment, be patient. Those will be done over the next couple of days and you'll have that grade. Remember, you have these 3 last lecture quizzes: vaccination, epidemiology, and the pathogen spread lecture. Don't forget to hand those in. Those are 5 points each and every point adds up, right? And then the final big chunk of points for the course comes with that last lecture exam. And that's a week from Friday. Okay. Okay. Any other questions about what's left in the schedule, or any questions about anything that you've been looking at in lecture or things that you've been reading? Feel free to type it into chat for me. And we can talk about it. This last portion of the course, the immunology and the vaccination and the epidemiology, it's very different from the other units in this course, in that we're sort of moving back away from the specifics of microbial cells, the specifics of bacteria and archaea and other microbes. And instead, we're talking more about the impact that microbial organisms have on our society. The immunology material really focuses in on how our bodies react when they are infected with a pathogenic microbe. The vaccination material that you may have already looked at talks about what vaccines are and why we vaccinate. What does a vaccine help us achieve, basically, as a community? So this last unit is much more about how we interact with microbes as human beings, and particularly how we interact with pathogenic microbes. Oh, Brooke is asking about our lab meetings, our Zoom meetings that are left. After today, Brooke, there's only one more meeting, and that's a week from today, the 28th. So the same day as the deadline for late work. And on that day when we meet, a week from today, we'll be talking about the vaccines. We'll talk specifically about the different vaccines that are now available to us to be immunized against the coronavirus. All right, so I'm going to pull up a slide that we saw several weeks ago when we first started talking about the coronavirus, just to remind us what this viral particle looks like before we start talking about these testing processes. So if you recall, coronaviruses all have the same basic structure, and you're looking at a diagram of that structure on this slide. These are spherical viruses. They are enveloped. The nuclear material or the genetic material is RNA, not DNA. And this virus has very characteristic spike proteins coming off of the surface. And I know you all have seen drawings of this virus more times than you would have liked over the past year and a half if you consume any news media. These spike proteins have gotten a lot of attention because these proteins are both how the virus gets into human cells and the target of the vaccines. So these spikes, these are proteins. These are folded up three-dimensional molecules that jut off the surface of this virus. And it's these spike proteins that are able to bind to receptors on human cells. And if you recall, the type of cell that this coronavirus gets into in our body is called an endothelial cell. So that's a special type of epithelium that lines blood vessels. So this virus, which we originally thought was a purely respiratory virus, as many coronaviruses are, turned out to be a vascular virus because it infects the cells again that line our blood vessels. And that helps explain a lot of the strange symptoms that people develop when they come down with COVID-19. It can infect the lungs. It can cause a viral pneumonia, but it can also really infect any other part of the body. And it has caused symptoms as far-reaching as gastrointestinal signs, vomiting and diarrhea, for example. Some people have experienced rashes in the skin. They've experienced kidney problems. They've experienced cardiac problems and a whole range of symptoms. So this is a very complex virus because it can infect a cell type that is found throughout the human body. Your body has blood vessels all through it. That's how oxygen gets delivered to your cells. And this virus is capable of entering into those endothelial cells and then causing an immune reaction. And it's the immune system in its effort to destroy the virus that is causing many of the symptoms that people experience, the fevers and the terrible chills and the muscle aches. Remember, those are not caused by the virus. Those are caused by the immune reaction to the virus. So what we want to talk about now today is what we've done in terms of testing for this virus. what we are able to use in terms of technology in order to know whether or not a person is infected with this SARS-CoV2 virus. So I'm going to pull up another set of slides for us now. Let me get this one out of the way. Okay. So we're going to start off just by talking about diagnostic testing very generally. So the kinds of diagnostic tests that we use for viral infections. And then we'll talk a little more specifically about the testing we have available for this particular coronavirus. Remember, viral infections are very, very common. And most of the time, we do not know what virus is infecting us. That kind of shocks people when you tell them that. For example, when you get a cold, if you develop symptoms of what we call an upper respiratory infection or a cold, if you go to the doctor with a cold, the doctor is likely to tell you that it's a virus that is infecting you. But there is no one virus that causes symptoms of a cold. There are many of them. There are viruses that cause diseases throughout the body. And while we know the identity of many of those viruses, there are many others that we do not know their identity. Viral infections can cause diseases that range from very minor, like that very annoying common cold, all the way to life-threatening. Now, one of the big problems around identifying viruses is that we cannot simply culture viruses in the microbiology lab the way that we culture bacteria. We can't do it. We've been talking all semester about how we take bacteria or patient samples that we think have bacteria in them, and we inoculate culture media. And we grow bacteria, don't we? We do it all the time. And then once we've grown them, we can test them for various things and we can identify the bacterium. We can't do that with viruses. Now, why is that? What is it about viruses? If you want to type it into the chat for me, what is it about viruses that makes it so that we cannot simply take viral particles and put them into culture medium and put the culture into the incubator and grow them? What is it about viruses that makes them so hard to culture? Yeah, Brooke has it. Brooke has it. You can't grow them in culture medium because they're not cells. Viruses aren't cells. Viruses are particles. Remember, they are not alive. Good job, Cheyenne. They're not alive. They're not cells. They don't have a membrane. They don't metabolize. They don't respond to stimuli. They are particles. That's all they are. It's particles of material that are able to bind to receptors on our cells. And it's that binding interaction that allows them to get in. And once those particles are inside of our cells make copies of them. We are the incubator for viral particles, like the coronavirus. So you can't just put them into culture medium. The only way to grow viral particles in the laboratory is to first grow the host cells in culture and then infect the host cells with the virus. Now that may sound fairly straightforward, but I can promise you it is not. Culturing bacteria is actually pretty easy. As we've talked about this semester, you just have to feed them a few key nutrients, carbohydrate and protein, some water, some minerals, and they'll grow for you, most of them. But it turns out that eukaryotic cells, like human cells, are hard to grow in culture. It's difficult to grow them. And that's why we don't routinely make viral cultures because it takes a lot of time and it's difficult. It's difficult to do it. It's difficult to grow the host cell and then infect it with the virus. So we're already behind the 8 ball with viral infections because we cannot just take the viral particles out of a nasal swab and inoculate some culture medium and grow them. We can't do that. We have to find other methods to do that. Okay. So what do we have available to us regarding viral testing, diagnostic testing for viruses? What do we have available to us in everyday life, in other words? There are certainly plenty of laboratories in the country doing research that are growing eukaryotic host cell cultures and then infecting them with viruses. But that is not routinely done in diagnostic laboratories, in hospital laboratories or commercial laboratories like Quest or LabCorp. They are not doing that kind of diagnostic culturing. There are what we call indirect tests, very non-specific tests for viral infections. Those are simply things like, what does the patient have for signs and symptoms? What are they complaining about? What do their vital signs show? What does their blood work show? There are signs that a physician can see when the patient's blood is analyzed, not viral particles. Remember, you cannot see those under the microscope, but things like how many white cells does that patient have right now? And what kinds of white blood cells are very prominent right now? What kinds of white blood cells are very low in the patient's blood right now? Those are the kinds of signs from the blood that a physician can use to help diagnose a viral infection. And of course, imaging is helpful. By imaging, I mean things like doing x-rays, doing ultrasounds, doing MRIs, doing CAT scans. We're taking an image of something in the patient's body. For example, when you do a chest x-ray on a person who has a COVID pneumonia, that pneumonia has a pattern in the lungs that looks different from other kinds of pneumonia. So when a patient comes into a hospital and has symptoms of COVID-19, one of the things the physician can do is order a chest x-ray and look for that patterning that tells the physician this is probably the coronavirus causing this pneumonia. Again, it's not specific, is it? I haven't shown this virus to be in that patient's lungs. I've shown an indirect indication, a non-specific indication that this person has the virus in their body, in their lungs. So indirect testing has been used for a long, long time. As I said a moment ago, it's just not feasible right now to directly identify viral particles from a patient. You need a special kind of microscopy to do it, electron microscopy, if you're trying to actually visualize viral particles in a patient sample. And even if you were able to access an electron microscope, let's say you were able to take a sample out of that patient's lungs, and for some reason you had access to an electron microscope, just looking through the microscope, you're not going to be able to know what viral particle you're looking at. It's very, very difficult to differentiate one type of viral particle from another. And of course, that's because, as we've discussed, there aren't that many variations, are there? There just aren't. There are naked viruses and there are enveloped viruses. The naked ones, they're polyhedral shaped or they're helical shaped. The enveloped ones, they can be spherical with spikes coming out of them. There aren't that many variations. So a person looking under an electron microscope, again, which is not commonly done, but if you were to look under an electron microscope, if you saw a spherical enveloped virus with spikes coming out of it, you don't know that that's coronavirus. There are lots of spherical enveloped viruses that have spikes coming out of them. So we just can't do it. We cannot take a patient sample into an ordinary laboratory, a hospital laboratory or a commercial laboratory, and directly identify the virus that is infecting that person. So what the heck do we do? Because obviously we can in certain circumstances diagnose someone with a specific type of virus. You may have been tested at some point in your life for influenza. If you've ever gone to the hospital or even to the doctor's office with severe flu-like symptoms, you may have had a test done for influenza. There are several tests that are available to physicians for specific viruses. They're just not direct tests. They're not a test that involves us seeing the virus and identifying it. So what are our options? Well, we can test for the presence of that particular virus's genetic material. We're going to talk about how we do that today. We can test for the presence in the patient of some viral antigen. We can also, as I said earlier, test for the presence of antibodies against that particular virus. So these are the three mechanisms of testing that we have available to us right now in ordinary diagnostic laboratories. Now, on this slide, you're looking at an electron microscope image. It's been colorized so that we can see this spherical viral particle. This happens to be the patient sample, which is why it looks all strange in the background. There are cells in the background here, and the viral particles are in and among those cells. I put this slide up because I want you to remember that seeing a viral particle like this under an electron microscope, that does not tell you what virus this is, because there are lots and lots of spherical viruses, enveloped spherical animal viruses that have spikes coming out of them. I've listed a couple of them down here on the bottom. There is the coronavirus, the SARS-CoV2 coronavirus that we're dealing with. Just a reminder, if your audio is on, go ahead and mute that for me. The influenza virus looks just like this. Herpes viruses look like this. Norovirus looks like this. Poliovirus looks like this. West Nile virus looks like this. You cannot just look and know what you're dealing with. You have to do some more testing. So let's talk first about antibody testing. Why in the world would we test someone for antibodies against a virus like SARS-CoV2? Well, we do it because we know that after a person is infected with a virus for a certain amount of time anyway, their adaptive immune system is going to produce antibodies against that virus and any other pathogen that they get infected with. We can identify specific types of antibodies. Remember, the human body and other animal bodies make antibodies that are specific against one particular pathogen. We often refer to antibody testing like this as serological testing. You may have heard that term. And one of the nice things about serological testing or antibody testing is that we can quantify it. Remember, we can get what we call a titer when we measure antibodies. We can count them. That's going to give us information as well. It's going to help tell us whether or not a patient has been recently infected or whether or not they were infected sometime in the past. So antibody testing is actually quite helpful. But remember, not all antibodies are the same, right? There are different types of antibodies. And when it comes to viral infections, we are specifically talking about IgG antibodies, IgM antibodies, and IgA antibodies. So we have to know which antibody to look for. And we have to test specifically for that. So it's not a simple testing mechanism, but it is available to us in diagnostic labs. Now, let's take a minute and think about why antibody testing might not be particularly helpful. What are the limitations to us of testing a person for antibodies in order to diagnose them with an infection such as COVID-19? First of all, the production of antibodies varies depending upon the pathogen, right? We talked about in the immunology lecture that some pathogens are more antigenic than others. Some pathogens just elicit or stimulate a stronger immune response than other pathogens do. The same is true of viral pathogens. Some viruses stimulate a very strong immune response, and some viruses don't. Previous exposure plays a role. If a person has been previously exposed to a virus, they're going to have a past experience with making antibodies against it. And for the viruses that we're able to get over and over and over again, think about influenza, for example, your previous exposure is going to matter. The health of the individual matters too. You have to have a healthy immune system in order to generate an immune response that's going to produce antibodies. If you are a person who is quite sickly, if you are a person who is very, very young or very, very old, you are not going to generate a strong immune reaction. That's just biology. Little infants, generally infants under the age of about 6 months, don't generate the same immune response that kids do as they get older. Their immune system is still developing at that age. They can do it after an age of about 6 weeks, 4 to 6 weeks. An infant can start generating antibodies on their own. They just can't do it as well as, say, a 5-year-old or a 10-year-old because their immune system hasn't finished developing yet. The same is true for elderly people. People who are in their 70s and 80s and 90s, because they're aging, their immune system is just aging as well. It's not that they can't make antibodies, but they don't make antibodies as well as they used to. So, for example, some of you may have heard recently that some people who have been fully vaccinated against the SARS-CoV2 virus, particularly people in nursing homes and long-term care facilities, some of them have come down with COVID-19, and they've come down with a reasonably bad case of it, although none of them, well, actually, I shouldn't say that. I think there have been a couple of deaths. So people are very confused. They don't understand, how could my grandmother have gotten her vaccinations and then come down with COVID? And part of the answer is that grandmother may not have been able to have a sufficient immune response because of her age. She probably made antibodies, but she probably didn't make enough antibodies to protect her from the virus. That's a function of her age. So there's lots of things to think about when it comes to antibodies. The other thing that's very important to remember about antibody production is this. It takes time for the human body to make antibodies. It actually does not happen until the later stages of infection. And we talked about this in the immunology lecture as well. The first immune reaction that occurs is not from the adaptive immune system, it's from the innate immune system. So the first part of the response to a viral infection comes from the innate immune system. And it's not until that is underway that the adaptive immune system gets brought in. And once the adaptive immune system becomes involved, then antibodies begin to be made. So we can't use antibody testing in a person who is in the early stages of infection. In other words, if they develop signs of COVID-19 and they come into the hospital, we can't test them for antibodies because their body has not had time yet to make the antibodies. We also know that the virus that causes COVID-19 can create asymptomatic infections in people, or some people are what we would refer to as asymptomatic carriers. This is a very unique feature of this virus. This virus created many more asymptomatic infections than other viruses do. We still don't understand why. We still don't understand why so many people were exposed to the virus, became infected with the virus, but showed no signs. Those people were still spreading the virus to others. And they didn't know they were because they didn't have any signs of illness. It turns out that in asymptomatic people, and this goes not only for this virus, but for other viruses, people who are asymptomatic don't tend to generate very many antibodies. And that probably makes sense, right? The immune system doesn't respond nearly as vigorously in those people because they're not sick. They don't need protection in that moment from that pathogen. Their immune system will respond, but it doesn't make as many antibodies as, say, a person of the same age and the same health status who is very sick with COVID-19. That person is going to generate more antibodies. So you have to think about that too. Antibody testing has limitations. Okay, so how do we test for antibodies? There are a couple of different ways that a person can be tested for antibodies, but one of the most common technologies is called ELISA, E-L-I-S-A. Now this stands for something. I'm going to type it down here on the slide for you. It stands for Enzyme Linked Immunosorbent Assay, ELISA. Now, there are lots and lots of variations of the ELISA technique, but the general idea is that it uses laboratory-produced antibodies and an enzymatic reaction in order to quantify viral antigen in a patient's sample. All right, now I said there were a couple different ways to do ELISA. We're going to talk first about the indirect method because this is the one that involves antibodies. So follow along on this picture with me and see if it makes sense to you. What the test involves is a container, that's what they're showing you here, that comes from a commercial kit. So you buy these kits commercially. The bottom of the container has been coated with the viral antigen or one of the viral antigens. So for example, for the SARS-CoV2 virus, it's part of the spike protein. This container is coated with this protein. It's stuck to the bottom of the container, in other words. Now, what happens in the laboratory is the patient's blood sample is separated out into cells and serum or plasma. It's the plasma that's going to contain the antibodies. So what we do is we spin the patient's blood, we take the serum sample, and we add it into this container. Now, if that person has antibodies against this virus, what's going to happen is those antibodies are going to stick to the antigen in the bottom of the container, aren't they? That's what antibodies do. If this person has these antibodies that are directed against the SARS-CoV2 spike protein, they're going to adhere to that antigen. They're going to bind to the antigen in the bottom of the container. Now, we then wash out any of the serum that is in the container. We just wash it right out and get rid of any extra material. So all that's left in here now is the spike protein that was there to begin with and patient's antibodies that are stuck to the spike protein. Next, what we do is we add in a reagent that comes with the kit that contains antibodies. And these antibodies are very interesting. They're not directed against the antigen. They're directed against other antibodies. Let me say that again, because that's important. Remember from lecture, we talked about how all antibodies have this Y shape. We said that there are two FAB regions. Those are the arms on the Y. And then there's an FC region. That's the stem, if you will, of the Y. The AB regions, the arms, are what differ from antibody to antibody to antibody. So the arms are only capable of binding to one particular type of antigen, such as the spike protein in this example. But the stem part, that's the same for all of the antibodies in the patient's body. That's the constant, the FC region of an antibody. So all we need to do in the test is add in some antibodies that were made in the laboratory that are able to bind to the FC region. And that's what you're seeing here. The arms have stuck to the spike protein if the patient has these antibodies present. We're going to add in a second antibody that can stick to the constant region. And these purple antibodies that were made in the lab, they have a little substrate attached to them. That's the little circle. And the reason they have that molecule attached to them is because we can add in an enzyme. Remember, it's called enzyme-linked immunosorbent assay. We can add an enzyme in, and it will turn this substrate a color. We're going to get a color change. The color is going to vary in intensity based on the number of patient antibodies that are in here. This is a quantitative test. If the patient has just a few antibodies, then those few antibodies will stick to the antigen in the bottom of the dish, the spike protein. And when we add in the laboratory-made antibodies, they'll stick to those few patient antibodies. And when we add the enzyme in, we'll get color change, but we won't get much color because there aren't that many antibodies in there. So we'll get a light color. A person who has a ton of antibodies in their blood, all those antibodies are gonna stick to all of the antigen in the bottom of the dish. And when we add the enzyme in, we're gonna get a strong color change in that sample. So what we do in ELISA is We take that dish after we've added the enzyme in, and we let a computer tell us. This is all computerized in 2021. We let the computer tell us just how dark that color change is, just how intense the color change is. And the computer will then put a value on that for us. It will tell us which colors correspond to which quantity of antibodies in the patient's blood. And of course, if the patient doesn't have any antibodies, there'll be no color change, will there? If there's no antibodies in the patient's serum, there's nothing to stick. There's nothing to stick to the antigen in the bottom of the dish. So the laboratory-made antibodies, they have nothing to stick to. And when we add the enzyme in, there's no reaction. There's no color change. So ELISA assays not only can tell us if a patient has antibodies, but how many antibodies they have. And again, that's called a titer, an antibody titer. You need to have a certain number of antibodies in your blood against any pathogen in order to be protected against that pathogen infecting you again. All right. So that's what we call an indirect ELISA. This is a test that's looking for the patient's antibodies. Does the patient have these blue antibodies? Not really, but does the patient have these antibodies in their blood serum? I just want to take a minute here to remind us. I've used this term antigen several times now. Remember, an antigen is simply some molecule or some portion of a molecule that is capable of stimulating an immune response. That's all it is. Antigens are typically proteins. It's proteins that cause most immune reactions. And the specific regions on the proteins are called epitopes. Spherical enveloped viruses like the SARS-CoV2 virus have these spikes on their surface, and they also have other proteins sometimes on the surface of the envelope, on the surface of this gray coating. Those proteins are going to stimulate an immune response. They don't belong in our body. This is foreign. This is foreign protein. And our immune system is going to respond to it. So it's these red proteins, and there are some other ones that are yellow colored in this image. Those are the proteins that are the antigens that our body produces antibodies against. So remember, the innate immune system acts first. It's the innate immune system that's on the front lines. And when we get infected with SARS-CoV2, it's the innate system that is going to attack it first. The innate system is non-specific. It's going to attack anything, any pathogen, any foreign material that penetrates through one of our epithelial barriers, whether it's up in our nose or back in our throat or down in our stomach or our intestine or our urinary bladder or our reproductive tract, it doesn't matter, or our skin, any place that a virus or another pathogen penetrates an epithelial barrier, that's going to set off an immune reaction. Remember, it's the PAMPs and the DAMPs, the pathogen associated and the damage associated molecules that are going to bind to the PRRs that we talked about on the sentinel cells of the innate immune system and set off that inflammatory response that we talked about in lecture. Remember, those sentinel cells, the macrophages and the neutrophils and the mast cells and the dendritic cells, those sentinel cells are the soldiers on the front line of your immune system. They're going to get everything started off And it's after the inflammatory response is started that the adaptive immune system comes in and you start getting antibodies produced. Now, in addition to being able to test a person to see whether or not they have antibodies against the coronavirus, we can also use ELISA, a different kind of ELISA, to see if they've got the virus. This is the test that most people have been dealing with this past year if they've had one of these rapid tests done. Now, we said a minute ago that in order to test somebody to see if they have antibodies against the SARS virus or any virus, you have to have a blood sample. Okay, so the swab going up your nose, you can't use that to test for antibodies. If you want to test for antibodies, you need blood. And you need to spin the blood so that you get the serum away from the rest of the cells. And you test the serum for the antibodies. If you're going to look to see whether or not the antigen is present, not the antibodies, but the antigen itself, the virus itself, that's different. Then you can use any type of tissue sample or body fluid sample that the virus gets into. And the reason that they put that swab up your nose is because that's where the virus first comes into us. The virus comes into most of us, most people, by being inhaled, either through your mouth or through your nose. And it gets into the back of your throat, your pharynx. And we've discovered through trial and error that one of the places that we can find the most virus in an infected person is in the very, very back part of their nasal passages. I know, I've had the swab done. I know, it's awful because they are putting that swab all the way back as far as they can go in your nasal passages. It's a very tender area. Okay, most things don't touch back there, but that's where the viral particles tend to be concentrated when they first come into our body. So if you want to test a person for the antigen, in other words, for the virus itself, you have to go where the virus is. And in humans anyway, at the first part of an infection, that's the back of their nose. It's the back of their nasal cavity. So the ELISA is a very similar looking test for antigens. It's just that it's done a little bit differently. The steps are done a little differently. This time, instead of coating the bottom of the container with the antigen so we can look for antibodies, this time we're going to coat the bottom of the container with antibodies so we can look for antigen. So we coat the container with antibodies that were made in the laboratory against the SARS virus. And then we put the patient's sample from that nasal swab into the container. If the viral antigen, in other words, if the virus is present in the sample, it's going to stick to the antibodies. So it's similar to the other ELISA. It's just that instead of putting the antigen in the container, we just put antibody in the container. And if the patient's sample has antigen in it, has virus in it, it's going to stick to the antibodies. Again, you have to wash this out. And then you add in more antibody, but this time, this is an antibody that has been produced to stick to the virus. And this antibody, again, has a substrate attached to it so that when we add in an enzyme, we get a color change. This is ELISA technology, and that's the basis of the rapid test that you've probably heard of. You can get rapid test results on an ELISA in about 15 minutes. Sounds great, right? Sounds great. But have you heard anything about the rapid test that concerns you? Has anybody heard anything about how accurate the rapid test is? The Abbott test is the other name it goes by. Has anybody heard anything? Remember, yeah, you've got it, Brooke. The Abbott test, again, for antigen, is not very good. It's not very accurate. Uh-oh, that's a problem, right? Yeah. In fact, some studies on the Abbott test, and I'm saying Abbott because that's the name of the company that makes the kit to do the ELISA. That studies have shown that that test can be as low as 50% accurate. In other words, just because you get a color change, that doesn't mean that the person is necessarily positive. You can get false positives. You can also get false negatives on that test. That's not super helpful, is it? That test has been under a lot of scrutiny, and yet it's being used all over the country to test people for the virus and to tell them whether they're positive or not. So, accuracy really matters. And every test, even the gold standard test of polymerase chain reaction, every test has its limit in terms of accuracy. Right now, the most accurate test that we have against this virus is the PCR, the polymerase chain reaction. Remember what that test tests for. That test is looking for the presence of the viral genetic material. It's looking for the presence of the RNA that virus contains. And it's an amplification test. It's going to make millions of copies of that molecule if it's present in the patient's swab. PCR is considered the gold standard of viral diagnostic testing. And we have been using it a lot for the SARS-CoV2 virus. We've been using it a lot, and it's a good test. But even PCR isn't 100% accurate. It's a lot more accurate than that Abbott test is, but it's not 100% accurate. Here's something that people sometimes don't understand about viral diagnostic testing. There is no test that is 100% accurate. And what that means is that some people who are told that they are positive are not really positive. And some people who are told that they are negative are not really negative. That's just an unfortunate fact of diagnostic testing. And the public in general does not understand that. The public thinks if I get a positive test result back, I have a positive result. I have the virus. If I get a negative back, that means I don't have the virus. And unfortunately, that's not always the case. So, testing for this virus over the past year, not only here in the United States, but around the whole world, has been a bit of a mess, hasn't it? It's been a bit of a mess. And to be fair, it was a brand new virus. It is a brand new virus. And we had to develop brand new test kits for it. The PCR test is still the best test we have. And its accuracy is up in the 90s. So it's still a good test. It's a decent test. But here's what's recommended. If you take a PCR-based test, again, from the swab up your nose, if you get a PCR test done on a swab and it comes back negative, and you think you've got the virus because you don't feel well, the very best thing to do is to repeat the test, because no test is 100% accurate. The same thing is true of that Abbott rapid antigen test. If you get an Abbott test and it comes back negative, let's say, and you're pretty sure you're sick, go and get a PCR test done, because the PCR test is better. If you get a positive test on a rapid test and you're shocked because you're not sick, you may be a carrier, you may be an asymptomatic carrier, or you may not have the virus. You've got to get another test done, though. We don't trust the results of a single test 100%, because we can't. The tests just don't have that level of accuracy. The polymerase chain reaction test for SARS-CoV2 is excellent. It's got an accuracy up in the 90 percentile. The antigen test is poor. It's got an accuracy as low as 50%. I mean, you might as well just flip a coin, right? At that point, you might as well just flip a coin. Do I have coronavirus? Let me flip this coin and find out. It's just not a good test. So, what's the take-home message about diagnostic testing for viruses? It's not great. It's not great. We are much better at testing for bacteria. We're much better at identifying bacterial infections than we are viral infections. It's just the nature of the pathogen. And right now, while we're so dependent upon these tests so that we can try to control this pandemic, we're limited. We're limited by the tests that are available and the accuracy of those tests. It's unfortunate, but it's true. All right. Yeah, if you're not familiar with that terminology, false positive, false negative, it's just like it sounds. If you get a false positive, that means the test came back positive, but you're not really sick with the virus. That would be a false positive result. A false negative is the opposite. If you get a test back and it says you don't have the virus, and you really do have the virus, that's a false negative result. And we measure false positive and false negative frequencies for diagnostic testing because those really matter, don't they? Especially during a pandemic. We don't want people to walk away from a testing site thinking, oh, phew, I don't have the virus, when they really do. Just like we don't want someone walking away from a testing site thinking, oh my gosh, I have COVID-19, when they really don't. So we try to limit false positives and false negatives as much as possible, if we can. All right. Any questions? Any questions about these tests? Any questions about the ELISA test? Remember, there's different versions of ELISA. There's an ELISA that tests for antibodies. That's the indirect one. Then there's an ELISA that tests for antigens. That's called a sandwich ELISA. There's all different versions of it. Very, very helpful test, very useful test. But again, the accuracy is going to vary. It's going to vary, and the accuracy really matters. All right, that's all I have for you today. Remember, you're really going to want to take a look at your grades as soon as possible, so you know if any of the assignments that have occurred through the semester you have not submitted. If you have not submitted an assignment, it really is in your best interest to submit it, because you're going to earn points on it, and that's going to boost up your grade.